Welcome back to the deep dive. Today we're looking closely at something pretty revolutionary in conservation science.
Yeah, we're diving into environmental DNA, eDNA.
Exactly, it's the genetic material just floating around in water, soil, even the air.
And to really get a grip on it, we're focusing on a key review paper. It's by Matthew Barnes and Cameron Turner from back in 2015.
Right, the title is The Ecology of Environmental DNA and Implications for Conservation Genetics.
That's the one. It really lays the groundwork.
So our mission today is to unpack what this paper synthesized.
Yeah.
The authors basically argue that if eDNA is going to be more than just a neat trick, we need to understand the actual rules governing it, the physics, the biology. They call it the ecology of eDNA.
Precisely. So maybe first, let's just clarify what eDNA is.
Good idea.
It's genetic material shed by organisms. But the key is it's collected from environmental samples like water or soil. It's not like finding a feather or scat.
So extraorganismal DNA, taken from bulk samples, not chasing the actual animal.
Exactly. You're sampling the environment itself.
And the significance here is huge, right? It gives us this unprecedented way to detect species.
Yeah, especially when finding the organism directly is, well, hard or maybe even impossible.
That phrase they use really captures it. Conservation in a cup of water.
It does, and that's why conservation folks jumped on this so quickly. Think about invasive species.
Ah, yes. Early detection.
Right. If you can spot an invader when there are only a few around before they really take hold.
You save so much trouble and money down the line.
Definitely. The paper mentions those classic examples like American bullfrogs in France.
Or the big-headed carp surveillance in the U.S. Midwest, those huge river systems.
Crucial early work. And they also talk about zebra mussels, African jewelfish, even Burmese pythons. Finding those first few is where eDNA really shines, much easier than, say, dragging nets everywhere.
And it's not just invasives. On the flip side, it's just as important for species that are rare or threatened.
Exactly, where you don't want to disturb them, or maybe they're just incredibly elusive.
Like the great crested newt in the UK.
Yep, or the eastern hellbender, that big salamander. Or endangered bull trout out west. Traditional surveys. They often miss these low-density populations.
But eDNA can pick them up. That sensitivity is key.
It really is.
Okay, so we see the need, we see the potential, these early successes, but the results weren't always consistent, were they?
No, not initially. And that's why this Barnes and Turner paper is so vital. It helps explain why results can vary. It gives us this framework.
The 4 domains to help interpret the data properly.
Right. We needed to move beyond just, wow, eDNA, to asking, okay, how reliable is this signal right here, right now?
And the framework is origin, state, transport, and fate.
That's it. Understanding those 4 is crucial.
So let's start with origin. Basically, where's this DNA coming from?
We know it's things like shed skin cells, mucus, feces, maybe even whole cells sometimes. The exact physiological sources. Still being worked out for many species.
What's interesting is how the organism's biology plays into it, like slimy creatures.
Yeah, amphibians, fish. They seem to be detected really well. It suggests mucus and body fluids are major ways the DNA gets out there. Being slimy is an advantage for eDNA detection, apparently.
Huh. And it's not just about size either, is it? More biomass usually means more DNA.
Generally, yes. Like with Idaho giant salamanders, they found that link. But age matters. Ontogeny. They found with bluegill sunfish that the little ones, the juveniles, actually shed more eDNA per gram of body weight than the adults.
More per gram. That's counterintuitive.
It is, but think about the implications.
Right. If you only sample when tons of juveniles are around, say, after spawning.
You might think there's way more total biomass than there actually is compared to sampling later. Your signal is strong, but your inference about biomass could be off.
That's huge for designing how and when you sample.
Absolutely critical. And it gets even more complex. They mentioned shedding rates can increase with certain behaviors, like feeding activity in those big-headed carp.
Okay, so that's origin. What about state? What form is this DNA actually in? It's not all just neat dissolved strands.
Nope. It's a mixture. Some might be inside intact cells, intracellular. Some might be free-floating extracellular DNA. And the size of these bits matters a lot.
Why does size matter so much? For filtering.
Exactly. Think about how you collect it. You're usually filtering water. A study on common carp found their eDNA in all sorts of particle sizes.
Okay.
But the biggest chunk, the highest percentage, was in the 1 to 10 micrometer range. So if your filter has pores bigger than 10 micrometers...
You could be losing most of your signal. It just washes right through.
Precisely. But if you go too small, say 0.2 micrometers, trying to catch everything.
Their filter clogs up instantly with algae and other gunk.
Right. It's this constant balancing act. Sensitivity versus actually being able to filter enough water. Understanding the state helps you pick the right filter pore size.
Makes sense.
Yeah.
Okay, next up, transport. How does this stuff move around?
This is key for figuring out what a positive signal actually means spatially. Is the fish right here, or was it upstream?
I remember the paper mentioning DNA tracers moving like salt tracers in streams, so water current is a big driver.
Initially, yes. But the DNA doesn't just flow forever. It interacts with the environment. It sticks to things, absorption.
Right.
And it can travel pretty far. They mention microbial eDNA from invertebrates being detected something like 12 kilometers downstream.
12 kilometers. Wow. So yeah, a positive hit doesn't mean the creature is sitting right under the boat.
Not necessarily. And then there's vertical movement, too. It sinks.
Right. It accumulates in the sediment.
Yeah, studies found high concentrations of fish eDNA down in pond and river bottoms, which means it settles out of the water column.
And if it settles, maybe it can get stirred back up. Which leads us to the last domain, fate. How long does it stick around? Persistence.
Exactly. If old DNA gets resuspended, it could look like the organism is currently present when it's long gone.
So how long does it last?
Well, DNA isn't super stable chemically. Degradation starts pretty much right away. And how fast depends on everything. Light, oxygen, pH, temperature, microbes, enzymes. It's really variable.
How variable? Like between land and water?
Oh, hugely different. On land, in soil, especially bound to clay particles, some DNA, like from GMOs, can last for days, maybe even years.
Years, wow.
But in water, especially with sunlight, UV, it's often hours to days for it to break down in the water column itself.
Hours to days is quick.
Very quick. That common carp example again. In an aquarium, the detection probability dropped below 5% in just 4 days. But
the sediment difference.
Exactly. In the pond sediments, they could still detect that same carp DNA over 130 days after the fish were removed. Gone from the water, but lingering in the mud.
Okay, this really ties it all together for practical use, doesn't it?
It does. The biggest challenge is telling apart a strong signal, because there are lots of organisms nearby, versus a strong signal because the DNA just lasts a long time or traveled far.
Fate and transport are crucial for figuring that out.
Which is why Barnes and Turner emphasize that mastering this whole ecology, origin, state, transport, fate is essential if we want to move beyond just presence, absence.
And the tech is moving fast. It's getting cheaper, more accurate.
Absolutely. Which leads into the future frontiers they identify.
Right. We're going beyond single species detection now.
Yeah, into whole community description using high-throughput sequencing, or HTS, often called metabarcoding.
Metabarcoding. So instead of one test for one species,
you use general primers that amplify DNA from, say, all fish or all insects in a single sample. You get a snapshot of the entire community.
That's a massive jump in efficiency. Imagine screening for hundreds of species at once.
It makes big biodiversity surveys much more feasible, assessing plant communities, soil arthropods from leaf litter, all from one sample.
Okay, what's next after community description?
The really big goal. Estimating organism abundance, moving from is it here to how many are here.
That's the quantitative leap.
Exactly. If we could reliably link eDNA concentration to population size or biomass, that would be transformative, especially for rare species you can't easily catch and count.
And there's been some promise there. Correlations found.
Yes, for carp, amphibians. Studies show correlations, but making it consistently reliable. That's tough because of all those ecological factors we just discussed, origin, fate, etc.
It always comes back to those 4 domains.
Pretty much. But as we understand them better, the quantification gets more robust. And beyond abundance, people are pushing into population genetics and genomics.
Using eDNA for genetics, how does that work?
Well, most eDNA work uses mitochondrial DNA, mtDNA. It's abundant, relatively stable, easy to find.
Okay.
But for finer detail like telling hybrids apart or estimating effective population size, you often need nuclear DNA. And that's much less common in eDNA samples, degrades faster, and it's mixed up from potentially many individuals, so it's challenging.
I see. It's a signal-to-noise problem almost.
Sort of, yeah. But the potential is there.
What about finding DNA in weird places? The paper mentioned alternative environmental samples.
Oh, yeah. Researchers get creative. They've found eDNA in snow from urine to detect canids.
In snow?
Yep. Or saliva left on twigs where deer or elk have been browsing. Even collecting DNA off spider webs or carrion flies that visited carcasses.
Spiderwebs as DNA collectors. That's brilliant. Non-invasive, too.
Totally. And then there's the tech side, remote and autonomous systems.
Robots doing the sampling.
Basically, yes. Systems developed mostly for marine microbiology can now filter water, extract DNA, even run the tests, all autonomously, way out at sea.
Wow. Sending data back by satellite.
You got it. And think about drones collecting water samples from remote lakes or dangerous rivers. It opens up sampling at scales we couldn't imagine before.
That's incredible.
Yeah.
Okay, one last frontier mentioned. Looking beyond DNA.
Right. Isolating molecules other than DNA, like environmental RNA.
RNA, isn't that less stable than DNA?
Generally, yes, but that instability can actually be useful.
How so?
Think forensically. Because RNA breaks down faster, detecting eRNA gives you a much tighter time frame. It suggests the organism was present very, very recently.
So DNA might say it was here sometime in the last week, but RNA says it's likely here right now, or was within hours.
Exactly, a sharper temporal signal.
Okay, so tons of exciting potential, but we need to be realistic about the challenges too, right?
Absolutely. The paper is clear about limitations. False positives, false negatives are always a concern. PCR errors, sequencing mistakes, issues with assigning DNA sequences to the correct species in those big HTS data sets.
You have to be careful.
Very careful. And it circles back to that core challenge. Is your signal strong because the source is close and abundant, or because the DNA just stuck around for a long time or traveled far?
Origin, state, transport, fate. You need to consider all four to interpret the signal correctly.
That's the take-home message from Barnes & Turner. Understanding the ecology of eDNA is, as they put it, integral to advancing these frontiers. We need that fundamental knowledge to make the applications reliable.
So, for you listening, the key takeaway is that eDNA isn't just a cool lab technique anymore. It's actively changing how we monitor ecosystems. But using it responsibly means understanding these underlying processes.
Yeah, and these methods are already being used to inform real-world management decisions. Invasive species control, endangered species protection. The science in this paper is crucial for making sure those decisions are based on solid, well-interpreted data.
Okay, so here's a final thought to leave you with. We talked about eDNA persisting in sediments over 130 days in that pond example. And we know scientists pull ancient DNA from things like ice cores or deep lake mud. So, if DNA can last potentially centuries or millennia down there, what does understanding the long-term fate of ancient eDNA mean for how we define what's native or alien to an ecosystem, especially as climate change shifts species ranges? That's something to chew on.